So I'm in the chemistry and biochemistry department. Let's see if the pointer is down here. No, now I know what you meant. Yeah, so I'm here in the chemistry and biochemistry department. And the story I'm going to tell you here is the result, or was recently, it's in press in the Journal of Biochemistry and Molecular Biology Education. This is the actual title, but it's really about something that came up yesterday with Carl Wyman. I don't know, is Carl here? Ah, there he is, okay. Which I thought was pretty interesting. It's neat to see people who are that positive about a challenging area. Okay, so let me see here. So I'm going to take you through just a course or a set of courses which I did this experiment on. It's Biochemistry 142. It's a 3-quarter series. It's upper division. It's required for the majors in chemistry and biochemistry. Typically, we have 100 to 150 students in there. I started the major with a couple of other people about 25 years ago. It's now bigger than the chemistry major. It's pretty challenging. I designed it very much because I was very aware of what was needed in terms of biochemists coming out. It's got 3/4 of labs in biochemistry and biophysics. There are a lot of electives, and a big fraction of those students do undergraduate research. So what was I trying to solve when I got this started? I was trying to solve some attrition problems, attendance problems, performance problems, because this class is extremely challenging, a lot of memorization in many examples from textbooks, et cetera. And I also wanted to focus on this here, which is one of the more difficult ones, which is higher level understanding of the material, experimental design, et cetera, things like that. So what have I been doing? So in 2008, I got started doing things that were pretty incremental. And this is sort of a timeline going from the beginning down to 2018. Started things like iClickers, learning goals, online quizzes, post-exam counseling to the people who do pretty poorly about how they're studying. Now, the main point, though, about what I do, or this got started about 2 or 3 years after this, was I created small groups, which I designed. I mentioned here composition and in-class positioning. What I mean by that is that I figure out the low-performing students from prior classes and put them together with higher-performing students so that and they don't know this, and so that there's groups of 3 or 4 people, and usually the higher, or the prior classes are organic chemistry and Chem 1 and bio. And then I also put them around the room in different places, move them around different times, because in a lot of these lecture classes, I need to be able to walk around and interact with them, and I can't if they're in the middle of the room. Now, the other part of this, many of you are familiar with a lot of this stuff, is online short answer and free response questions, which are posted. And then in the next few slides, I'll explain some of that. Basically, I spend most of the time in that classroom actually talking about those questions. There are no more, right around here, I started giving no lectures. So there was no lectures at all anymore. And I also do 2-stage exams, which Carl and the group that he's associated with have promoted, which I like. It's a really neat idea. Students like it too. And then the last thing that I've been doing, the last couple things, is I've been focusing on science process questions, which are kind of content independent. And then I've also started something that's sort of like our last speaker talked about, but not as elaborate, where there's seminar attendance, and I engage those students in what those seminar speakers talked about. Okay, so what does a typical week in my class look like? On Friday, I post online multiple choice questions and free response questions, which cover about 15 pages of material. On Monday, the students, Monday at 8 a.m., the students post their responses to those, and I can see their scores, and I can see what they're lacking in, et cetera. And the class is typically at 10 a.m. on that Monday. I then post the questions for Wednesday. On Wednesday, the students post their questions or their answers by 8 a.m., and it repeats itself. Okay? So I'm literally looking 2 hours or 3 hours before the class at what those students did. And that helps me guide what I'm going to be doing in that classroom. I obviously have a lot of materials prepared ahead of time before I come to that classroom, but that guides what I focus on. So a typical day is students walk in, and there's a multiple choice iClicker question. And it's usually not about the content. It's usually about some kind of science process question, and I'll show you an example of that. And then I focus on the problematic areas. Now, if students do 70% or more on average on questions, I don't tend to focus that in-class time on those areas. I will have a variety of things that I can pick from. I have iClicker questions. I have multiple choice questions. I I have free response questions, and all these things I can pick from depending on how they did that, on their on the homework part of this. So free response. So, on the free response, what they do is they answer the questions on a 3x5. They put the date and their name on it, and then they turn those in. Now, what happens is that a big part of this whole thing is, and I can't convey this enough, those of you who've done anything like this know what I'm talking about, a big part of this is right through here, where the students are given, not in every case, but in many cases, a chance to talk among themselves, and then I call on groups to give their answers. I listen to their logic. If their logic is compelling, I tell them so, and if it's not compelling, I ask somebody in the classroom to comment on that. Okay, so what they're hearing is other people comment on the logic and the illogic of their interpretations of what they just saw. And that's a pretty big part of what I do there. I typically have 2 to 5 eye-clicker questions, 2 to 4 free response questions. And there's not a lot of lectures, but if you popped in any one day, you might see me use 2 or 3 slides of a lecture. Okay, so this is going to go very quick because I don't have the time to go through all these things. Here's a typical list of the learning goals that I use, which are typically 5 to 15 for any one chapter. And you can see how it's framed. After completing chapter 6, you should be able to classify an enzyme based on the reaction that it carries out. Okay, and the students really have to focus on those things because that's what I'm going to be using to guide the design of the exams. Okay, so here's a typical experimental design question. I don't have the time to go into it. But basically, this is pretty much content independent, although you're going to see content. This is really about, can you think like a scientist? Can you interpret things? So basically, I give them some data. This was a seminar that was given here. Here's some data, and then some background. And at the bottom is the question, which is stuck. Okay, so the question is, are you convinced by this data? And then there's some possibilities, and then they have to be able to justify that. So that's what I meant by process questions. So this is the kind of stuff that I focus those kinds of questions on. Again, it's a variety of things. What's a testable question in science, et cetera, et cetera, et cetera. I don't have the time to go over all these things, but you can see there's a lot of different things here that are broad across science. It's nothing about biochemistry, really. Okay, typical eye-clicker question, which is just multiple choice. This happens to be from a chapter on enzymes. Here's a typical short answer question, which has the learning goal that is being focused on associated with it. Here's the answer, which the students see as well. This is the focus here. Okay, so how did I design this experiment? I had one active learning classroom, that was me, and I had one TA. The main point of this, what I was trying to do here, was to see whether it's possible, which is why the title was what it was, one professor, so I'm not a lecturer, I'm a professor, do research, and one TA, can you pull off this kind of thing? That's what I was asking the question of, okay? How much work does it take to do that? And I was comparing myself to 2 experienced biochemists, and the students self-selected which path they wanted to go on. We found that there was no bias in the academic performance of the students coming into these 2 different paths, and there were multiple choice questions. Now, I don't give multiple choice exams, okay? But these 2 instructors did, so I used their exams, okay? And there was also final exams, and the grading was done blind. Okay, so the performance was, here's the active learning, it's in blue. Here's the conventional. So there's a 3-quarter series. We did it for 2 quarters. This is the first quarter. So my class was the blue. You can see it skews to the right. These lower performing students here were moved to the right, and there's also movement over here as well. That was for 142A. Oh, and the averages were 65 and 54, which is pretty significant improvement. This is the box plot for the individual midterms, final, and here's the total over here. Same thing for 142B. This is the second quarter. And again, active learning was blue, conventional lecture was red. You can see it skews to the right, and the average is to the right. Again, okay, so the grading was done in this way, which was basically online quizzes and short response, midterms, eye-clickers, in-class short response. So what did I learn? Okay, so the attendance improved, not surprisingly. The attendance improved significantly, okay? Because remember, one of the things I wanted to address was attendance. So this is a typical number for many, many science classes on our campus, and my attendance on average was above 90%, okay? The content performance improved. That's what I just showed you. What I'm not showing you is that the students improved very significantly. This is much more difficult for me to judge because other instructors don't tend to give, other instructors I could compare to don't tend to give those kinds of questions, but the students improved significantly from where they started to where they ended up in terms of their ability to identify when an experiment wasn't designed right, when it was designed right but interpreted wrong, when there was a problem with precision versus accuracy, how do you deal with outliers, the ethical questions about dealing with outliers, all that kind of stuff. Okay, so now here's the question. This is kind of what I was getting at with when we heard the talk yesterday. Why is change so slow in the face of such compelling evidence? The evidence that's been published by Carl Wyman and his group and the paper I'm just referring to, it's overwhelming that this kind of stuff works. So the question is, why isn't it more broadly brought in? There's a lot of really creative things being done here, but I'm talking really about full professors who are doing all this cool research. Why aren't they doing this kind of thing? Okay. So I think the answers are here. I took a page from Carl Wyman's talk here. That's the big one, and these guys get smaller, okay? So the big one, I think, is incentives. Most of the colleagues in my department look at me and think I'm an oddball. It's just weird. Here I am, NSF, NIH funded, and what am I doing this stuff for? This doesn't help my science focus. It doesn't help me get promoted faster. I'm a full professor for 15, 20 years. It doesn't matter that much. But the fact is that the incentives aren't there, okay? And a lot of the things we're talking about, they're great, but unless you get the big body count of full, actual research active professors doing something different, it's going to be the lecturers and those kinds of people that are doing these things, okay? Okay, this is a kind of a humbling thing. I think there's an amazing number of smart people in science education, and they've invented many, many good things, ways of doing things. Unfortunately, it doesn't work like it does in science. If somebody publishes a new PCR, or it's 1985, somebody publishes a PCR method to amplify a gene, I don't need to convince the molecular biologist in here to use it. It works. You do it. There's no argument. In science education, and I think it's because you're dealing with people, there's always an argument. There's always well, maybe Carl Wyman's really charismatic, or maybe Norbert Reich is really good, or whatever. It's not just the data. And I find that very perplexing and frustrating as a scientist because I can show people publications that say this works, yet it's not being incorporated, okay? Anyway, I think it's because you're dealing with people. And then there's, of course, I think the big problem here is infrastructure. I'm talking about why doesn't it get implemented more broadly. You saw the number of hours some of the people put in. I put a lot of hours in as well. I get comments from my students saying, my God, the guy doesn't even teach. We do all the work. It's really kind of a trip because they don't see the underneath of it all. Anyway, and then the final one that I love when I get this from, usually they're chemists, some physicists. Norbert, it worked for us, the other way. Why do we have to change it? And I'm like, wow, horse and buggy, cars. Anyway, that's it, I think. Let's see. Yep. Yeah, Steve.
On the online quizzes or the in-class, well, for the online ones, are they told if their answer's correct?
Oh, yeah. Yeah, I didn't have the time to do all the kind of nice stuff that a lot of people before me here did today, but there's a lot of feedback. There's go to this page. This is where the answer's given. They actually get 2 tries on some of the quizzes. There's a lot of stuff I do there that's not as elaborate as many of the people here do the online stuff because I don't want to really be online. I want to have FaceTime with these people in that classroom. But you're right. I mean, I do give that feedback. Yeah.
So we have this barrier issue on our campus as well, where a lot of faculty don't want to teach in the lower division, and many of them are voluntold that they need to. And so what we've gone to is to remove the onus of any development from their having to teach. And we have a dean that supports them getting full credit for teaching for 2 weeks of a 10-week quarter. And the second time they teach for 5 weeks. And our goal is to give them the learn by doing experience. So they practice actually answering a student's question in class.
But you're just talking about trying to get senior faculty to teach, period, right? Or you're talking about... Oh. But you're talking about teaching something like this. Oh, I see, I see. Right, so this, right. So this was brought up by Carl as well in a different way, and that was there's incentives like give me money to go do something, and then there's incentive, which is like a one-off thing, and then there's incentives like, oh, I'm up for a review, I'm going to get promoted, and you know what, I did all these teaching things, and that counts. And this will sound very cynical, but my view of education at the UC system is that when you go up for promotion, it can hurt you, okay, but it hardly ever helps you, okay? Okay. What? That's great. I'm a guarded optimist. I mean, I think eventually it'll work. I just don't want to. I'm not going to be around for 200 years, so I just don't want it to be. I'd like to see it move a little faster. So I think that's encouraging. But to me, it's like, dude, look at the evidence.
Okay,
I get into some serious fights with some of these people because they're like, it's denial. It's like fake news,
So
yeah.
let's have one more only.
Carl.
A quick comment is that you have to keep in perspective. It was 40 years between the time when somebody did the experiment of having doctors wash their hands with the patients and saw an enormous decrease in deaths. Sure. And it was 40 years before that became hospital policy. So you have to recognize people are a little slow to shake.
Yeah, okay, so like I said, he's an optimist. Okay.
I'm sorry to cut it off, but you can certainly ask Norbert your questions during the break.